Well, thank you so much. My name is Kim Nye, and I am the president and founder of TESS Research Foundation, which aims to find treatments and ultimately a cure for SLC13A5 deficiency, which is an epileptic encephalopathy. I'm speaking today about my family's personal journey with genetic epilepsy. I think my family is a case study in the importance of early and accurate genetic testing. And I will also discuss what happens after diagnosis. Um, Genetics includes the study of heredity, so I figured I would start with a small family history. I was a really lucky kid. I'm one of 7 kids in a crazy, fun, blended family, as you can see from this picture. I'm down here at the bottom. My husband had an equally blessed childhood. This is a more recent photo, but he is one of 4 kids, all healthy. We have no significant history of any major childhood illnesses in our family. My husband and I went to high school together, and then we went to Princeton University together, and when we realized we couldn't seem to escape each other, we decided to get married. We moved to England, where we split our time between my graduate school at Oxford University and Zach's at London Business School. Life was pretty darn perfect, and after being married for a couple years, we decided it was baby time. 41 weeks into an uneventful pregnancy, I was induced, and 24 hours after being induced, my doctor decided that things were not progressing, and sweet baby Tessa was born by C-section. It was December 22nd, 2003, the same year that the Human Genome Project was completed. Tessa seemed to be perfectly healthy, but we couldn't get Tessa to eat, and she turned really blue during her first bath. On Christmas Eve, a doctor walked into my hospital room. He told me that Tessa was having fits and that she had most likely had a stroke. We would know more after an MRI. I had no idea what a fit was. It turns out it's a British word for seizures. I don't think I really even knew what a stroke was. I was definitely in shock. I remember delivering the news to my husband. My big, strong husband dropped to his knees and wept. We were 23 years old. After a couple of weeks, we were able to control Tessa's seizures with phenobarbital and phenytoin. Her MRI was normal, and as the months went on, she looked like a healthy little 6-month-old. She received her second diagnosis, benign idiopathic neonatal seizures. We thought we had escaped unscathed from a close call. We took Tessa off her seizure medications, and on the day of what was supposed to be her last dose of phenobarbital, she had a seizure. We were never able to control her seizures again. Tessa spent the next few years in and out of the hospital. She had dozens of hospital stays under her belt by the time she entered preschool. We knew our ambulance crews by name. Tessa tried dozens of medications in different combinations to no effect. We traveled across the country to see the best neurologists. We attacked the seizures logically. We tried standard anti-epileptic medications. We tried steroids and later IVIG in case it was an autoimmune problem. We tried pyridoxine and other supplements in case it was a metabolic problem. We had more MRIs and PET scans and EEGs to see if she was a surgical candidate. We implanted a vagal nerve stimulator and suffered through the ketogenic diet several times. We had every gene test available. We sought out researchers who could do exome and genome sequencing, which was not yet available clinically. Tessa's diagnosis shifted to catastrophic epilepsy. You can see in the highlighted section of this slide, at the bottom of this slide, that Tessa was having up to 53 seizures per hour. She did not have the genetic marker for Dravet syndrome, but her clinic notes started to say things like, her epilepsy phenotype most closely resembles Dravet syndrome. We weren't sure she would survive until kindergarten. We still could not find an underlying cause for Tessa's seizures. The one thing we took solace in was that none of the doctors we saw thought that Tessa had an inherited genetic epilepsy. We were told to be fruitful and multiply, and we complied. Lily and Maggie were born uneventfully. They are wonderfully healthy and normal. In 2012, we received news that Tessa had an ATP1A3 mutation, alternating hemiplegia of childhood. My husband and I were tested. We were both negative. AHC is almost always a de novo mutation. We celebrated, and a year later, we decided to have another baby. When I was 7 months pregnant, We found out that an error had been made in the lab. Tessa's sample had been switched with another child's. Tessa did not have alternating hemiplegia of childhood. We were back at square one, but we were still hopeful that our fourth child would be healthy. On August 23, 2013, I gave birth to a seemingly healthy baby boy named Colton. My husband and I knew our family was complete, and we were confident that we were the luckiest people on the planet. But at around 2 a.m, Colton started having trouble feeding. He started looking blue. I remember calling in the nurse and telling her something was wrong. She assured me that Colton was fine, but I suspected that Colton was having seizures. Doctors agreed to transfer him to the NICU for observation. Around 5 a.m, I emailed Dr. Brenda Porter at Stanford. I had only met her twice before, but despite the early hours, she responded to my email within minutes. She had a neurology team in place almost immediately. Colton was indeed having seizures. I went from dreaming of future college tours and picking out football pads to knowing that my son would likely never talk or live independently. Dr. Porter put Colton on the medications that had been the most successful for my daughter, Tessa. It took us 4 years to find out that Diamox had helped Tessa, but Dr. Porter started Colton on Diamox when he was 3 days old. Whereas Tessa was in and out of the hospital for years, Colton came home from the hospital at 2 weeks old, and he has remained amazingly stable. We immediately reached out to every researcher and geneticist that Tessa had ever seen. We sent samples of Colton's blood to the NIH Undiagnosed Diseases Program, to Baylor, even to the doctor who had misdiagnosed Tessa with alternating hemiplegia of childhood. None of them could find a genetic marker. When Colton was 3 months old, a friend introduced me to Matthew Bainbridge. Matthew is now at Rady Children's Hospital, but he was a genetics researcher at Baylor at the time. We talked for quite a while about the kids and our family history, and when I hung up the phone, I remember wondering if I would ever hear from him again. A few months later, I got another call from Matthew. He said, "I think I found the gene, and I think there is something to do about it." Matthew had found compound heterozygous SLC13A5 mutations in Tessa and Colton, a citrate transporter disorder. I introduced Matthew to Brenda Porter. We tried potassium citrate to no effect. We measured citrate levels in the blood and urine and CSF. We tried various supplements. Matthew and Brenda reached out to a drug company that was developing a medication called triheptanoin. An agonizing year later, we had FDA, hospital, and drug company approval for a triheptanoin trial. I flew with my kids to Baylor to start the miracle oil. My kids gagged down for months. No miracles occurred. We started hearing about more and more children diagnosed with SLC13A5 mutations. They all had the same symptoms: intractable mixed epilepsy beginning shortly after birth for no apparent reason, persistent seizures of multiple types resistant to medications, severe difficulty with speech production with better receptive language, movement and coordination problems, episodes of body stiffening or weakening, brain MRI that appears normal or has subtle changes in the white matter, tooth enamel abnormalities, and mildly elevated citrate levels in the blood. Tessa continues to have many seizures daily. Most of them are short, like in this video. You okay? Yeah. But Tessa likes to throw longer seizures at us as well. Here's a tonic-clonic seizure.
Tessa, Mama's right here. You're gonna be okay, sweets.
This goes on for many minutes, but I think you get the idea. She is globally delayed, has minimal language, and she staggers when she walks. I always try to remember that second diagnosis, benign idiopathic neonatal seizures. Unfortunately, that is not our reality. I wish I could say that Colton is perfectly fine. He is not. Although we have almost completely controlled his seizures, he is now 4 years old with minimal language, and he is just starting to take independent steps. but I do feel confident that he benefited from having a big sister who had spent a decade trying different treatment options. Having an early and accurate genetic diagnosis is like giving every child a big sister. It lets families and doctors know that they are dealing with a serious epilepsy. So that is my family's journey to diagnosis, and I'm going to shift to talk about life after diagnosis and a little bit about where we are today. I figured I'd start with my advice for families on diagnosis day, or maybe you can look at it as what a doctor might think about saying to a family on diagnosis day. I think the first thing that I wanted to hear was that I didn't do anything wrong. The reason I highlighted what a blessed childhood I had and the amazing education that my husband and I were both able to receive is to highlight that genetic epilepsies can happen in good, smart families. It's not the parent's fault. I would also tell myself on diagnosis day that my child is the same person today as he or she was the day before diagnosis. In the end, diagnosis doesn't change that much in your day-to-day life. Your days are still going to be filled with physical therapy and speech therapy, and hopefully with birthday parties and play dates. I would tell myself, don't wait on a cure, but a little hope is not a terrible thing, and definitely connect with other parents. You will find that you are not alone. And to help your child find interests and hobbies. I think that it's hard when you realize that your child is not going to have the regular milestones in life. They're not going to go from high school to college and have a career, but they are still a little person with interests, and developing those interests becomes a big goal, I think, for parents who are on this unexpected journey. I spent a decade hearing other families' epilepsy stories and feeling like they were so different from my family's journey. It was a lonely and isolating experience. I think early and accurate genetic diagnosis helps families avoid a diagnostic odyssey, and it gives them a sense of community. We have a really close community in our SLC13A5 deficiency group. If you want to move on to a little action after a diagnosis like we did, I think that there are some simple and free or inexpensive steps that you can take that help families find each other and help doctors and researchers find families and really create those bridges between the various groups. So we set up a website, a Facebook page, and added a link to our website on the SLC13A5 Wikipedia gene page. Usually that Wikipedia gene page is the first thing that pops up when you Google the gene. So it gives you some really good search engine optimization if you can put a link to your website on that Wikipedia page. We started connecting with SLC13A5 families around the world. They would simply Google the gene. That's what every parent seems to do. And even doctors and researchers do that. And it helped us all find each other, and it still continues to work. We didn't know if there was a treatment that helped consistently because this was such a newly described disease. And so Dr. Brenda Porter created a questionnaire for the families, and it asked about treatment options that they had tried. Some of the other groups have had real success finding things that did work or didn't work. For our group, it wasn't as obvious, but it's still, I think, a necessary next step to figure out, what everybody has tried and whether there's a consistent medication or treatment to continue on or to avoid. For our group, it became clear that we needed a more formal way to organize our disease community and to collect funds for research. So we formed TESS Research Foundation. And then we assembled the dream team to figure out where the gaps in research were and to review research grant applications. So Brenda Porter is the treating neurologist for my two children who have the disease, and she has been with us since day one. And Matthew Bainbridge is the researcher who found the mutations in my kids, and he continues to stay with us too, which is amazing. And then we assembled, more people who we thought could really weigh in on where these gaps in research were and had experience figuring out where the best next steps might be. We came up with 3 priority areas. Number 1, understanding the mechanism of how the dysfunctional citrate transporter protein causes seizures and other neurological problems. Number 2, create disease models that replicate the human phenotype. And number 3, in terms of treatment options, preclinical screening of candidate treatment molecules or therapies, and clinical trials of new or existing therapies. And then we announced a research funding opportunity. In our case, we awarded $100,000 grants, and we gave out 3 grants last year. And you can see our 2017 grantees who are working with human neurons, flies, mice, and zebrafish models of SLC13A5 deficiency. If we examine the progress from when the gene was first described as pathogenic in July 2014 until today, I think there are some major breakthroughs. I'm going to jump down sort of to this third bullet point. Testing is much easier. Rather than requiring whole exome sequencing, we can now diagnose SLC13A5 deficiency through epilepsy panel tests. We also have metabolic screenings that have that suggest this disease, and a citrate biomarker, since the kids have elevated citrate in their blood and CSF. We know now, this fourth little bullet point, that the known pathogenic mutations result in a loss of function. And while we do not have a cure, we do know more about treatment options. Several children have had seizure improvement with acetazolamide or Diamox, which I mentioned in my family's journey. And we have some hypotheses about NMDA receptors with this disease. We know that triheptanoin, which was suggested in the first publication, actually did not help the handful of kids who tried it. There are also stories of the ketogenic diet being not helpful or even harmful to some of the kids with this disease, which was a surprise. And we're cooking up plans for 2018. We're working on biobanks and gene therapy and patient-derived iPSCs and better expression studies. And none of this would be possible without a genetic diagnosis. So in terms of the research, I think it is critical to diagnose these genetic epilepsies. I wanted to end with a little recap of why genetic diagnosis is important, even if there isn't a current therapy, because while there are examples like GLUT1 transporter that have clear treatments with a genetic diagnosis, a lot of these newer genetic epilepsies that have genetic markers don't have a current suggested treatment. But I still think it's important to diagnose them. And I guess for me, these are the top few reasons. First of all, parents can better predict the future. They can find camaraderie and they can learn from other families. Families can consider reproductive implications. In the case of SLC13A5 deficiency, it's an autosomal recessive disease, which means that the parents have a 1 in 4 chance of having a child with this disease if they're both carriers. So that's some important information for our families to have. Families can enroll in clinical trials now and in the future. They can share clinical information with other researchers. They can discover treatments that have worked and not worked as they become available. And they can decrease the testing and searching for other etiologies. Hopefully, more and more families will have an end to their diagnostic odyssey. I think those are the main points that I wanted to touch on. I'm happy to take any questions that people have, but thank you so much for listening to our family's journey and to learning a little bit about TESS Research Foundation.